So one technique we use in our lab is to measure ongoing electrical activity using electrodes that are placed on the scalp. And what we've discovered in the last year or so is that if we look at the rhythmic activity that is coming out of the brain, that is neural activity in a specific frequency.
Electroencephalography. So basically we measure the electrical potential that the brain uses. So the neurons in the brain communicate with tiny bit of electricity. And we try to pick it up and amplify it in an amplifier, and then we can look at it. And the idea behind it is that if you do the same thing with the brain, then the same kind of pattern will appear. And if you do it 1,000 times, all the noise will just go away, and you'll have like a clear idea of where in the brain. And this one's particularly about when something happens, right? It's very timing specific. You can measure at millisecond accuracy when something happens in the brain.
The electrodes on the scalp of this subject that measure the ongoing electrical activity while they're engaged in memory and perceptual tasks. And this allows us to understand how that electrical activity changes depending on what they're perceiving and what they're holding in mind. Then by measuring that activity's distribution over the scalp, we can actually decode what the content of their memory is when they're holding an idea in mind. And this gives us some insight into how rhythmic activity is used to generate and support memory and attention in the human brain. Now, with our collaborations with our systems neuroscience colleagues, they have the opportunity to actually look inside the brain of animals that are also holding memories and that are also perceiving similar kinds of visual information. But here, they get a much more detailed view of how the brain activity is evolving at the cellular level, and they can even manipulate that brain activity using laser light in genetically altered animals. And these kinds of techniques give us a much more powerful view of how those brain areas are actually supporting memory and causing the maintenance of these kinds of ideas while the animals are performing different types of tasks. So the hope with the animal research is that by allowing us to manipulate brain activity at the cellular level, we can get a much more detailed understanding of how specific brain areas are supporting specific psychological processes like memory and perception. And this kind of information is akin to looking under the hood of an automobile. If you can see the engine at work, you're going to have a much better ability to repair it, to improve it, and to understand problems when they arise. So the human research is important for establishing the relevance of a particular brain phenomenon with things that we care about, like how effective is memory, how effective is attention, how are these types of mental abilities affected in different clinical disorders. With animal models of these processes, we hope to get better information that will help us to understand exactly what neural circuits generate these psychological processes, and that will give us information for how we can diagnose problems, rehabilitate problems, and otherwise kind of engineer the system to operate more effectively.